You float. You've been floating for some time now. Minutes? Hours? You cannot tell. There is no hunger because you are not quite alive. There is no cold because you are mostly just shaped a tiny head, a thin tail, delicate fingers reaching for something you cannot name yet. You float through dark water. Around you, other things. Bits of broken cells. The occasional bump that might be another phage like you, might be nothing. You do not think about this. You cannot think. You only exist. And then, touch. Your tail fingers brush against something huge. A surface, rough with bumps and valleys. You recognize nothing consciously, but your body recognizes everything, the shape, the chemistry, the rightness of it, like a key sliding into a lock. The walls of a bacteria, your bacteria, the ones you were built for. You stick. The attachment is permanent. Your tail contracts. Like a syringe pushing down, your genetic material, the instruction manual that makes you flows through your hollow center into the warm inside of the cell. Your outer shell stays behind on the surface like abandoned clothing. Everything that matters about you is now inside. The bacteria keeps working, unaware. Its tiny machines build proteins. Its DNA copies itself. Its walls pulse with the normal traffic of life. But you are there now. Something happens chemically, automatically, like water running downhill. You have heard, though hearing is not the right word. Though you do not hear anything, you have sensed, perhaps, in the structure of what you are, that some phages choose fire. They activate immediately. They turn the cells into a factory within minutes. They exploded outward in a cloud of copies, 40 minutes from entry to explosion. Fast, violent, immediate. You did not choose this. You choose to wait. Your DNA does something quieter. It finds the bacteria's main instruction book, that long loop of heredity, and cuts into it. Your genetic material slides itself into the host's genes, settling between them like a bookmark between pages. You become part of the bacteria. You stop being separated. You are now part of its inheritance. When it copies its DNA, it copies you. When it divides into two cells, both daughters receive a copy of you, sleeping and integrated. You pass down through generations. 2 cells become 4, become 8, become thousands, and in each one, you waiting. The bacteria lives. It eats sugar, builds proteins, swims through its environment. It does not know you are there. Or perhaps it does, perhaps your presence changes it subtly, protects it from other phages like you, gives it advantages you will never see. You wait. Time passes. Many generations. The bacterial colony grows. You exist as information, as potential, as a ghost in the machine. But the world is not stable. The world fights back. The bacteria begins to change. Perhaps in the next generation, perhaps the 10th. Its surface bumps shift, random accidents, survival pressure, evolution speeding up around you. The lock your key was made for begins to change shape. Other phages float past the bacteria now. They attach, but their grip fails. The lock has moved, or disappeared, or hidden itself behind a wall of blocking proteins. You are inside, integrated, but your siblings, if you had siblings, if copies counted as family, cannot enter. They floated away, unsuccessful, their tail fingers grasping at surfaces that no longer welcomed them. You sense this only distantly in the changing chemistry around you. The bacteria is learning to resist. And then, stress. The environment shifts. Perhaps ultraviolet light damages the DNA. Perhaps the food vanishes. Perhaps antibiotics attack the cell walls. The bacterium's emergency response activates repairing proteins flooding everywhere, trying to fix what is breaking. These proteins recognize you. Your integrated DNA cuts itself out. 1 cut. 2 cuts, you are separated again. And now, activation. The bacteria becomes a factory. New phages assembled inside. The cell swells with copies of you. The cell bursts. You emerged into an environment you did not enter. The descendants of the original bacteria surround you. You infected them. Some you destroy immediately. Some you enter and hide inside, beginning the cycle again. Your patience has multiplied your possibilities. But possibilities are not promises. You float through new territory now. The environment has changed. You are no longer in the comfortable liquids where you first emerged. Perhaps you are in food now, acidic and thick with chemicals. Perhaps you are in a stomach, burning your acid. Protein shells. Perhaps you are in water, but the water is hotter than before, or the saltiness has changed, or sunlight streams down from above. Your shell begins to break down. Slowly at first. A protein here, a bond snapping there. The perfect shape of your head loses its perfection. Your tail fingers, those delicate sensors, melt in the heat. You are still mostly intact, still theoretically infectious, but each moment wears you down further. You float towards a bacterium. Contact. But your tail fingers are damaged now. They stick poorly, slide off, fail to trigger the injection. The bacteria drifts away, indifferent. You remain outside, empty and waiting. You float. The sunlight finds your DNA through the protein shell. Your genetic code corrupts, instructions becoming gibberish. Even if you enter a cell now, you will not copy correctly. You will produce broken proteins, incomplete genomes, phages that cannot assemble. You are breaking. And then, perhaps, you find another cell. Your damaged tail somehow attaches. Your DNA, corrupted but still partly functional, injects into the cell. But this bacteria is ready. Its immune system, its memory of past infections recognizes you. Special proteins bound to your DNA, guided by archived records of phages like you. They cut you apart before you can hide, before you can activate, before you can become anything at all. You are destroyed within seconds. Or perhaps you enter a different cell, and this one has no memory system, but the human body, because you are inside a human body. Now, inside the gut of something vast and warm, this human body has defenders circulating, antibodies. They recognize your shell proteins, stick to you, mark you for destruction. Immune cells swallow you whole. You dissolve in the acidic interior of a hunter cell, your proteins unfolding, your DNA fragmenting into pieces that will be recycled into something else entirely. You float, but you are floating towards dissolution. Or, you successfully entered a cell. You integrate. You wait. The bacterial colony grows, and you pass through generations, patient and sleeping. But the environment shifts again. The human takes medicine, antibiotics, broad and merciless. The bacterial colony dies. Billions of cells rupturing at once, their walls breaking, their contents spilling into the gut. You are released, but there are no hosts left to infect. The environment is empty now, or nearly so. You float through nothing. Days pass. Weeks. No contact. No attachments. Your proteins keep degrading. Digestive enzymes in the gut break down your tail. Your head cracks. Your DNA leaks out and is consumed, chopped into fragments too small to mean anything. You stop existing as a complete thing. Or, you persist. Against the odds, against decay, you find another host. But this host's surface has changed. Mutations. Lock modifications. Your tail fingers attached, but weakly. The bacteria produces cutting enzymes that recognize foreign DNA, ancient defenses, crudder than memory systems but still effective. Your genome is chopped apart before integration. You failed. You float again. Attached again. Failed again. The cycle continues, but with each try, success becomes less likely. Resistance spreads through the bacterial population. They share defense genes like warnings passed from cell to cell. The entire community adapts around you. You are becoming obsolete. You float through a gut ecosystem now, complex beyond comprehension. Hundreds of bacterial species, thousands of phage types, all interacting in networks you cannot understand. You attach to the wrong bacteria when your sensors almost recognize, but not quite. You inject your DNA into a cell that cannot support. You're copying. Your genetic material floats uselessly in foreign space, then degrades. Wasted. You float through bile, through slime, through the hostile landscape of living tissue. You encounter bacteria hidden in protective films, communities wrapped in shields that the tail cannot penetrate. You encounter bacteria not actively dividing, poor hosts for your strategy. You encounter bacteria that are simply elsewhere to deepen tissue, too far from where you float. Geography defeats you, or time defeats you. You float for hours, days, and your window of function closes. Protein shells are not eternal. DNA is not stable forever outside a cell. You become less infectious with each passing moment, until finally you are just debris, organic molecules waiting to be recycled. You end, not with a burst, not with the dramatic explosion of successful copying, but with quiet dissolution, a gradual return to parts. You stop being a phage and become merely atoms, scattered and purposeless. You float. Somewhere in the liquid around you, a bacterium carries a gene for antibiotic resistance. During your last copying, you accidentally packaged some of the bacterial genes along with your own. Now you carry that gene. When you infect the next cell, you might transfer it. You do not know this. You do not know anything. You are not a mind. You are a pattern that continues, a cycle that repeats, a message passed between cells in a language older than intention. You float and stick, and inject and copy and burst and float. Again, again, again. The cycle empties of meaning through repetition until there is only the rhythm. Float, stick, inject, copy, burst. Float, stick, inject, copy, burst. Floating stick, inject, copy, burst. Floating stick, inject, copy, burst, float. You have no memory of the first time. You will have no knowledge of the last. Somewhere, the shell breaks down completely. Sunlight shatters your DNA. An enzyme tears open your head. No host appears. The particular shape of your tail fingers finds no matching lock, and you disintegrate and named into the background noise of molecular chaos. Or you do not. Or you persist. Or you continue the cycle until the last bacteria dies, until the last environment welcoming to your specific shape vanishes from the universe. You float. You've always been floating. You will always be floating. The story has no beginning and no end, only the eternal middle, the endless present tense of seeking and entering and bursting and seeking again.